but she classy. The type to suck your dick without asking. I like a bitch to pull up and get to action. Yeah. We don't play, yeah. We should just pray, yeah. We don't play, play. I don't want no girlfriend, but I think I'm feeling you, and I know you're feeling me. You gon' be alright on when you're with me. I don't.
Okay, so now we're gonna start with metabolism. So basically, what is metabolism? Metabolism by itself is a chemical reaction that occurs in a living organism, and the processes in metabolism involves in the conversion of food into energy in the form of ATP, the formation of carbohydrate, protein, lipid, and nucleic acid. So now we move to type of metabolism in a cell. So there are two types of metabolism in a cell, which are catabolism and anabolism. So catabolism is a process of breaking down the complex substances into simple substances, and this process releases energy. For example, the breakdown of glucose during respiration, during cellular respiration to generate energy. And anabolism is the process of synthesizing complex molecules from simple molecules, and this reaction use or absorb energy. For example, the formation of glucose during photosynthesis.
Okay, so moving on to the next subtopic, which is 5.2, and that is enzyme. So in a cell, biochemical reaction occur at a high rate to protect the living process. The biochemical reaction can occur quickly due to the presence of enzyme. An enzyme is an organic catalyst that is mostly made out of protein and is produced by living cell organism. So the enzyme is called as an organic catalyst because it is mostly made out of protein, and that is for organic, and it actually speeds up a chemical reaction without itself being affected, and that is catalyst. The substance needed for an enzyme reaction is called a substrate. The substrate will combine with the enzyme at a specific site and form an enzyme substrate complex. Moving on, we got the enzyme nomenclature. In the 1960s, the IUBMB, International Union of Biochemistry and Molecular Biology, introduced the enzyme nomenclature based on the substrate or reaction it catalyzes. The name is derived by adding ASE to the name of substrate it's catalyzed. Some of the examples we got here are maltase, amylase, lactase, and lipase, and protease. However, there are also a few enzymes that do not follow this naming system, especially enzymes that were discovered before this naming system was introduced. Some of the examples are trypsin, pepsin, and rennin. So lastly, we got the general characteristic of enzyme. So which it is enzyme acts rapidly. Most of the reaction catalyzed by enzyme are reversible. Enzymes are only required in small quantities and are reusable, and the structure of enzyme itself remains unchanged and are not destroyed after a reaction.
Again, welcome back to Bio. Okay, today I'm going to talk about intracellular and extracellular enzyme. What is the meaning of intracellular enzyme? Intracellular enzyme is enzyme that are synthesized in a cell for its own use, which means the cell produce enzyme and use it for itself. For example, the hexokinase enzyme that is used in the glycolysis process during the cellular respiration, while extracellular enzyme is enzyme that are secreted outside the cell, which means the enzyme that is produced by a cell is secreted outside the cell to be used by other cell. Okay, for example, the trypsin enzyme is produced by the pancreatic cell and secreted into the duodenum to break down the polypeptides. I'm going to talk about the mechanism of enzyme action. What is the mechanism of enzyme action? An enzyme attracts substrates to its active site, catalyzes the chemical by which products are formed, and then allows the products to dissociate or separate from the enzyme surface. The combination formed by an enzyme and its substrate is called the enzyme-substrate complex that are shown in the picture here.
Assalamualaikum and good evening to everyone that watching this video. So today we're going to talk about a subject in biology. So we're going to discuss about the mechanism of enzyme. Okay, so there are many factors that affect the mechanism of enzyme, and such as temperature, pH, enzyme concentration, and also substrate concentration. First thing we're going to talk about is temperature. So as we all know, at low temperature, the rate of reaction catalyzed by enzyme is always low. But when the temperature rises, the kinetic energy of substrate molecule also increase and will increase the frequency of effective collision between the substrate molecule. Well, at optimal temperature, the enzyme is at its maximum. So after it reaches some maximum quantity of heat, it will stop at that, like that, stop. Okay. After reaching the optimal temperature, any further increase in temperature will reduce the enzyme activity. So we all know, the further the heat increase, the lower the enzyme activity rapidly. Okay, the next we're gonna talk about is pH. Enzyme activity is influenced by the pH of the surrounding solution. So most enzymes are most effective in the range of between pH 6 and 8. We all know 7 is a neutral, right? Okay. However, there are some exceptions. For example, pepsin enzyme in the stomach at the optimal pH range of between 1.5 and 2.5. So the change in pH value changes the charge of ion. Well, the extreme change in pH value will break the structure chemistry bond and change the activity site of enzyme. So that's all. Okay, next is substrate concentration. If the concentration of enzyme is fixed while the concentration of substrate is increased, the reaction rate controlled by enzyme will also increase. Remember that, okay? So when substrate concentration increase, the opportunity for an effective collision between the substrate and enzyme also increase. Well, at the maximum level, the concentration of enzyme become a limiting factor. The reaction rate can only increase when the concentration in enzyme increase. So, okay. Lastly, enzyme concentration. Okay, when the concentration of enzyme increase, the rate of enzyme reaction will increase because of the presence of more active sites. If the concentration of an enzyme in one reaction is double, the amount of substrate converted to products per unit of time is also doubled. So you have to remember, if the concentration is double, then the substrate converted to products per unit of time is also double. Lastly, at the maximum rate, the concentration of the substrate becomes the limiting factor, and the rate of reaction can only be increased by adding more substrates. So that's all for me. Don't forget to subscribe and like our video. Thank you. Bye.
So again, biology. We move on to 5.3, the application of enzymes in daily life. Please. Okay. Enzymes have long been widely used in commercial sector and everyday use. And what is immobilized enzyme? Immobilized enzymes are enzymes that combine with hidden and invisible substances to increase the resistance of enzymes towards change of factors such as pH and temperature. Okay. Next, there are 6 examples application of enzymes in daily life. The first is digestive enzymes used in medical sector. Second is lactose enzymes used in lactose-free milk. Third, protease enzymes are used to
separate the fish skin. Other than that, tricks and enzymes used to extract the animal sperm. Then we have pectinase and cellulase enzymes used in juice production, and last but not least, amylase, lipase, protease, and cellulase enzymes in biodetergent. Okay, that's all from me. Thank you. Stay at home. Don't stay in the house. Okay, bye bye.